నమస్కారం. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం విద్యార్థులకు స్వాగతం. ఈ ప్రసారంలో
And today's topic is clinical biochemistry, in which we are going to study how a disease is identified or detected and treated and other parameters are done. Clinical biochemistry is an application of chemical and biochemical techniques used to detect diseases. The clinical biochemistry labs estimate the chemical constituents of various body fluids, and the body fluids include the blood sample, urine sample, cerebrospinal fluid samples or tissue histopathology. When venous blood is collected, anticoagulants are not used, but when you are taking the arterial blood, and it should be collected by the physician, and that is only for the analysis of blood gases. And a serum specimen is mostly required for the biochemical analysis, and if plasma is required, then an anticoagulant is added, namely the heparin. Heparin is commonly used because it does not affect any chemical reaction, and it does not and it does maintain the pH. Apart from the blood samples, we also collect urine and cerebrospinal fluids, tissue biopsies, where the histopathological studies are done using a technique called microtomy. The biomolecules like proteins, lipids, carbohydrates are done by colorimetric method, and lipid profile is studied to study the heart diseases, and liver function test is done to undergo the liver diseases or to study the liver diseases. Renal function test is done to study kidney failures. Apart from all these things, enzymes are also analyzed in the laboratories. For example, the aldolase enzyme, it is detected when there is a problem in heart and when there is a muscular disorder like muscular dystrophy. Then how to collect this blood? Blood or any sample is collected in a clean container, and these containers should be clean and free from detergents and bleaching agents, and generally the syringe is used for collection of blood. After collection of all these samples, the routine biochemical analysis is done, and to estimate the quantity of these biochemical parameters, a technique called photometry is used. Then what is photometry? The photometry is based on the physical laws of light. In this method, the intensity of absorbed, transmitted, reflected or emitted light is measured and related to the concentration of the test substance, and this photometry is based on a law called as Lambert-Beer law. Now we have seen how the blood is collected and how it can be used for analysis of biochemical parameters. Two most important disorders, number 1 inherited disorder, and number 2 the metabolic disorders. Inherited disorders are many in number. Nearly 250 diseases are identified, and these are genetic disorders, and they are carried from one generation to another generation, from parent to the offspring. And these inherited disorders can be diagnosed by two important methods, namely the first one is karyotype method and second one is by amniocentesis. Karyotype is done in adult, whereas the amniocentesis before the birth or pre-birth analysis, and if it is in pre-birth condition, amniocentesis is the method where we use the analysis by collecting the amniotic fluid from the womb of the mother, and once the amniotic fluid is collected, it is centrifuged and then DNA structure is analyzed. Similarly, the same sample can be used for the analysis of karyotyping. By both these methods, we can analyze the change in the number of chromosomal structure or change in the number of chromosomes, etc. That means altogether a variation in the chromosome structure can be studied. Changes in chromosome number or chromosome structure can cause genetic disorders. Two major types of chromosomal changes can be detected in karyotype: a change in chromosomal number, a change in chromosomal arrangement. Here I am giving two examples, a monosomic condition and trisomic condition. In a monosomic condition, you find there is a loss of chromosome, that is 2n minus 1. In trisomic condition, you find addition of one extra chromosome, that is 2n plus 1. And this trisomic condition is clearly seen in a disorder called as Down syndrome, that is 47 chromosomes are observed. If you see the picture, this trisomic condition occurs 1 in 800 births, and trisomy occurs due to one extra chromosome at 21st chromosome. And you can observe some phenotypic changes in this disorder. The first one is the mental retardation, and second one is epicanthic fold in the eye and a large furrowed tongue. This is a karyotype where you can see the 21st chromosome has got one extra chromosome. This is called as trisomy and which can be observed in Down syndrome. Similarly, Turner syndrome, which is a monosomic condition where loss of one chromosome is seen. Instead of having 46 chromosome, we have 45 chromosome here, and only one X chromosome is observed. In this condition, the female sterility is observed. Klinefelter syndrome, which is 47 number of chromosomes is present, and here XXY chromosomes are observed. Individual males have some fertility problems, but few additional symptoms are also observed. From the karyotype, you can identify that extra X chromosome is present in the karyotype. And coming to the next disorder, hemophilia. Then what is hemophilia? Hemophilia is continuous blood disorder, or it is a group of inherited blood disorder in which there is a lifelong defect in clotting. And this clotting disorder is due to the lack of some clotting factors. Number 1, clotting factor 8, that which is termed as hemophilia A, and hemophilia B is clotting factor B, which is also called as Christmas factor. And here is a diagram where you can observe hemerythrosis in severe hemophilia condition, where you can observe red patches due to the continuous bleeding process. Coming to the third group of blood disorder, that is sickle cell anemia. Here is very interesting structure you can see on your monitor. Number 1, the RBC cell of humans, it is almost circular in normal condition and biconcave in its structure. And during this disorder, the person feels fatigueness and lot of joint pains, and the most important one is anemia, and even the patient can suffer from jaundice. Then coming to thalassemia. Then what is thalassemia? Thalassemia is also one of the inherited blood disorder that causes mild or severe anemia. Now, till now we have seen some inherited disorders. Now, let us see for the metabolic disorder. Metabolic disorder is a condition where you find the metabolism does not take place properly, or there is no continuous metabolic pathway formation. For example, we are going to take phenylketonuria. As the name itself indicates, phenylketonuria means the phenylalanine is excreted in the urine sample. This can be identified at the early ages only, or early stages of the life. Now, coming to the next congenital disorder, that is albinism. I think most of us, we might have seen albinism in and around us. This is because of lack of an enzyme or a lack of a chemical called as melanin. Underproduction of melanin leads to the graying of hair, and you find some grayness on every part of the body, and these people, they cannot move freely in daylight, and they can live happily in the dark conditions. Now, coming to the next disorder, that is Gaussian disease. It is also a genetic disorder. which affects almost every people, and this is because of an ineffective enzyme that which is called as glucocerebrosidase. Now, finally, we will go for the next disorder, that is gout. Gout is a disorder where you can observe the deposition of urate molecules. Urates are the metabolic end products of purine metabolism, and this gout is caused because of deposition of monosodium urate crystals in tissues. Here are certain diagrams where you can observe deformities in the joints of hands, elbow, etc., where you can observe lot of swellings, and this swelling is because of the accumulation of urate crystals. And this is the advanced condition of gout, where you can see severe swelling in the hands as well as in the joints. All this is because of the urate crystal deposition. The most important carbohydrate metabolic disorder is galactosemia. Galactosemia is also an inherited disorder, which is an inherited recessive deficiency in enzymes that metabolize galactose. And in this condition, galactose is seen in a higher level in the plasma, and this galactosemia occurs due to the deficiency of an enzyme which is called as GALT. GALT refers to galactose 1-phosphate uridyl transferase enzyme. When this enzyme is inhibited, the accumulation of galactose takes place. Here is a small diagrammatic representation where you can see the galactosemia, where the glucose is not converting to energy because of lack of GALT. The same thing can be observed in a child also in the infant stages. Due to galactosemia, the brain gets damaged and the child gets cataract, jaundice, enlarged liver condition, and all this is because of excess production of sugar which is not further converted into energy. And the simple test done for the diagnosis of galactosemia is a blood test and the enzyme activity of GALT in RBCs, and even the urine analysis reducing substances can be observed. And apart from this regular biochemical analysis, genetic tests can also be done by analyzing the DNA analysis. Then coming to the next type of disorder, which is almost related to the eyes, and this condition is called as glaucoma. Then what is glaucoma? Glaucoma is a progressive optic neuropathy with loss of retinal neurons and the nerve fiber layer. Here in this disease, the retinal ganglionic cells totally die or they are killed by the disorder, and all this is because of high IOP. IOP is internal ocular pressure. Because of the pressure exerted, the retinal cells undergo death. Here is a small diagram for you. The iris is almost protruding. The protuberance of the iris is because of the pressure created inside the iris. And how to analyze the glaucomic condition in a laboratory? There are different methods of measuring the IOPs. One is applanation, and the next one is tonopen method, and third one is non-contact method. I think from your diagram you can see the different instruments that which are used for the identification of glaucoma, and this is the tonopen method. Till now we have seen what are the different disorders and how to identify them in a clinical laboratories. Now let us shift to a new topic under the same clinical biochemistry as per your syllabus, that is organ transplantation. Then what is organ transplantation? Organ transplantation is taking a graft in the sense a cell, a tissue or organ from an individual who is called as a donor, and it will be pasted in the other organism or in another individual who is called as a recipient. And generally the different tissues like kidney, liver, pancreas, intestine and heart, and even today the lungs are transplanted. Then what are the different types of transplantation? Transplantation are of different types. Number 1, autograft method, isograft method, allograft method and xenograft. Autograft means, auto means self, self in the sense a tissue can be taken from one individual and it can be transplanted in his own body, and here the most important thing is there is no need of any immunosuppression, very important. And second one is isograft, where a tissue is transformed between a genetically identical individuals. And third one is allograft, where a tissue is transformed between genetically different members of the same species. Here the important point is immunosuppressive agents should be given or required for the continuous transplantation process. These are the two main important features of this organ transplantation. Xenograft, as it is a transferring of tissue between different species. Here we are concerned with two important features, one is the autograft method and another one is allograft. Autograft needs no immunosuppression, but whereas the allografts they require the immunosuppressions. Then let us see how this transplantation immunobiology works. Whenever you go for organ transplantation, the most common problem that which we come across is rejection, and this rejection should be controlled, and this rejection process is because of two important antigens. They are called as transplant antigens, and namely the major histocompatibility complex MHC, and next one is HLA. In humans, MHC is called as human leukocyte antigen or HLA, which is located on chromosome 6. HLA molecules can initiate a rejection and graft damage, and this is done by two methods, one is humoral method and another one is cellular rejection. Humoral rejection is mediated by recipient's AB group, and cellular rejection is a more common type of rejection after organ transplantation mediated by T lymphocytes. Then what are the complications that which we come across during organ transplantation? The basic problem that which we come across during organ transplantation is a clinical rejection. Graft rejection is a complex process involving several components including T lymphocytes, B lymphocytes, macrophages, cytokines with resultant local inflammatory injury and graft damage. Then how to control this graft rejection? The graft rejection can be controlled by giving some immunosuppressants, and these immunosuppressants they reduce the rejection process. This comes under clinical immunosuppression action. The success of modern transplantation is in large part because of successful development or effective immunosuppressive agents. Two types of immunosuppressants are given. One is by method of induction, giving drug administration, and second method is maintenance of immunosuppression by giving biological and non-biological agents. Biological agents include monoclonal and polyclonal antibody treatment, and non-biological agents like giving corticosteroids. And of course, these are some of the old methods of controlling the rejection, but the modern biology has introduced new methods like cell therapy and stem cell therapy. Of course, these two therapies are still in progress in research, but somehow these theories or these therapies can also be included under transplantation. Cell therapy is the use of living cells to restore or maintain or enhance the function of a tissue or organs. And stem cell therapy means in which the stem cells are induced. Then from where do we get these stem cells? We get these stem cells from two most important parts, that is from your bone marrow and another is from the caudal blood. And by collecting these stem cells, we can go for the repairing of certain tissue damage like muscle constructions and other cell structure constructions. But there are two more main important problems of these theories, because they exhibit immunological rejection problems and biological problems like they create teratoma and chromosomal abnormalities. Then what are the major organs that can be transplanted? The thoracic organs like heart and lung can be done. Other organs like a liver, kidneys are also transplanted. Then next, even the tissues and cell fluids can also be transplanted. Tissues like cornea and the islets of Langerhans hands can also be transplanted. This is something like a modern biology where we can repair the damaged parts of the body. Now, we will go for another new topic, that is biomedical engineering. As the name itself indicates, it is a combination or it is a hybridization of skills and knowledge, in which we are combining the biological study as well as the engineering technique. The use of engineering science and maths to tackle problems in medicine, and it mainly focuses on machines, devices or non-biological structures of research, and the study of biomedical functions of the human body using mechanics, electricity, magnetism, optics and thermodynamics. It helps in improving the medicine and saves the life. Then what are the different sub disciplines under this biomedical engineering? We have three different disciplines. One is cardiovascular technology, next is neural technology, and third one is orthopedic technology. Cardiovascular technology, which helps in diagnosis and controlling and treatment of the heart diseases or cardiac diseases using some machines. Similarly, the neural technology involves the brain and nervous system. Orthopedic technology involves the skeletal system. And what are the machines that which are commonly used in the diagnosis of all these diseases? The heart-lung machines, which replaces the heart and lung functions during the surgery. Similarly, we got different instruments for measurement of your heart functioning and brain functions also, like echocardiogram, ECG and EEG for brain disorders, etc. Here are certain instruments you can see on your monitor. Number 1 is heart-lung machine, which can replace the heart-lung function during the surgery period, and the next one is EEG machine, echoencephalography machine, which almost studies or analyzes the brain defects, and finally you can even use the MRI, magnetic resonance instrument, and ultrasound instruments. These instruments like for example the MRI can help you in detecting the structural deformities at a cellular level by taking slices of your tissue, and similarly the ultrasound imaging technology, which helps you in the changes or abnormalities or detection of some tumors or any other aspects by means of image.
మా కార్యక్రమాలపై మీ సూచనలు సలహాలు ఆహ్వానిస్తున్నాం. మీ సూచనలు సలహాలు మాకు తెలియజేయవలసిన చిరునామా: డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్గ్, రోడ్ నంబర్ 46, జూబ్లీ హిల్స్, హైదరాబాద్ 500033.